you guys have so many questions, you too. Say, if you see antibodies, is that disease there or not? So this disease that does not manifest until after virus clearance is the antibody, okay? If you see the disease is there and I don't see antibodies present, that means you don't have the disease, okay? That means you are not seroconverted. If you have a seroconverted means you definitely saw the virus or pathogen at some point, okay? That's always put in your brain, cerebellum, okay? Diseases that have long incubation periods, you should see the antibody. Detecting past infection, you should be able to see the antibody. Like if you have a dengue, probably if you check next year at some point you may have antibody, which should protect. I don't know if you have not done typing, then you can say, okay, it will protecting the type 1 or type 2 or something. But yeah, that's why people want to make sure that if you get reinfected, and we mentioned that, that it may aggravate, because we talked about, right? Antibody-mediated enhancement, which is very bad for dengue, which sometimes be lethal. And good thing, I just watched the news. So dengue, there is a new vaccine came out. So check it out in this thing. So in Brazil, it has been approved and it may roll out everywhere. I think the end next year. So another is IgM specific assays, which is mostly IgM. If you saw in a lot of virus, we talk about IgM comes first, IgG comes later. So that's why they say when it is a recent infection, you detect this. Later on, you see IgG. Nucleic acid tests are so these are the advantages. Just read it, or I'll just keep it. PCR and all those kind of assays are very sensitive, but too specific, you don't know. Amplification, those are things you have to make sure, you're doing PCR, icons are very sensitive, but is it specific? That's why you're asking multiplex and asking, oh, if there is something, that is your job, not mine. You have to make sure that the assay you are recommending is correct. There is no contamination, there is no anything false positive, false negative, that kind of thing. So that is more rigorous exercise you have to do on those to standardize. It's easy to obtain but difficult to maintain. Some enzymes expiry date. You have to make sure you're not using expired chemicals, not properly stored. It's multiple time freezing and thawing. You damage those reagents. So those are the caveats there. Okay. Antigen antibody not as sensitive or specific, but there are also chances of cross contaminations. So basically, every assay has a pros and cons, like limitations. So that's you have to consider when you run an assay. Like COVID, it has like a gold standard. People thought about like PCR because it is quicker, but also you have to make sure the lab where it's doing. Oh, it is coming from Apollo. Okay, maybe the name is a good people, but is the technicians are doing the right thing, right? You should confirm those, check it out. Oh, you can ask definitely, because you are paying for it. For COVID test, say, hey, show me your standardized data, your control data. You have the right to do because you are paying for it. And they have to provide those data. That then you are sure, that, okay, my test is correct. So that way you have to, because all assays has drawbacks, and you have to make sure you are covering those things. Even for ELISA, if you see IgMs, repeat it again, or send that sample to another lab. They are also seeing the same thing. So that way you're also verifying, okay, my assay is also same like what that person is doing. So that way also you verify. So also people use different reagents. Oh, I'm not saying this, my positive came good, but my test sample is negative. That means it's correct. But you also send the samples, blindly. Say, hey, I have two samples. Can you tell me which is good or bad or positive and negative? Then they can say, oh, this is positive. Then you confirm it. Okay, what I did is correct. So. Specimen collection is also very important. The quality of samples, and I think Dr. Ashok mentioned, like, collecting samples, especially for clinical samples. How soon are you collecting? How you are transporting those? So those are the very important part. How you're maintaining the cold chains, duration of collection and testing. How you're storing those samples. Those are all vital, and those all depends on how your diagnostic should be, right? Some guys took the blood. Okay, I'll always collect blood from home and I'll give you the report. Okay, he went there and eating idli somewhere and talking with somebody and went for a movie, and they said, oh, I forgot my blood is still there. I have to send the report, and it's already my 40-degree outside and in his scooty, right? He kept it outside, right? And then the report came. And do you believe it? So those are the things you should think about. So the another interesting, it's a very old one, but the tissue microarray. It is now I'm going a little different. So here we are looking at the tissues. Okay, so mostly people do biopsies. If somebody has a breast cancer or other cancers and colon cancer, always people take biopsy. Doctor take biopsies and measure it. So here is an example like. So this is actually sweet potato. You can use that also for making microarrays. The microarray means what? The microarray. Micro means small, array means you're doing multiple things, right? So for example, here you are collecting those biopsies. Like, if I have a colon cancer, the doctor might have taken multiple sites in the colon. Colon is big, the biggest, very long. That's why it's called large intestine. It's a part of large intestine. So they took multiple biopsies from the colon. And then you have multiple biopsies and then put it there and put in this tissue. Okay, it's a sweet potato, and put those tissues there and then fix that. The whole sweet potato, fix it with formalin. So all the things, organism, everything, even the potato, even the tissue, everything will fix. Now it is solid. Now you embed it with paraffin. Then it becomes a solid paraffin. Probably you have seen paraffin blocks or those things. Visit some lab, get, huh? Exactly. So you make a paraffin blocks and then you section it and then detect it and then do the same thing, immunofluorescent antibody, or maybe just H&E, just normal slide H&E, and you can detect if there is any change in the cell morphology, if you see any change in the more neutrophil influx or megakaryocytes or bigger cells, you can easily doubt, okay, there is a cancer cell there. That's the way pathologist does.
Why do you eat the potato?
It's a cheaper, and you can put so many tissues inside. That's the guy who discovered that way.
Okay, instead of making multiple paraffin blocks.
Because it's easy. Yeah, exactly. That's why it's a microarray. So you just make a slice of this, and you have a good chunk of everything, right? You have taken multiple pieces from different tissues, and you just stain the whole thing, and you can, under microscope, you can just read the whole thing. Rather than doing single. You can do single. Then you should not say as a microarray. It's a micro single assay or something. It's not an array anymore. So genotoxicity, it's very quickly I'll go. So this is mostly used for cancer research. People do like a substance, whether the substance is cancerous to the tissue or cancerous to anything. So here people a lot of do the MS test, and here you can do that that substance or chemical, and you have the tissues there, and then put it there, and then incubate, and then want to see if there is any possible mutagens there. If the cells changes behavior, if the cells are doing something, then you can say, okay, that substance contains some carcinogen. That is same like transformation, which you saw, like virus changes the cells, normal cell to transformation. It's same thing, like it's mostly used for testing any kind of pharmacological substances. They say before it goes to the market, always they test this. And also they can do the animal model, also the same thing. Whether an animal is dying, animal has a liver function is damaged, kidney function damage. That's mostly people look for in animal model. They put the substance and do the, kill these animals and check their liver and kidney. If they have a toxicity, they can say the liver damage or kidney damage. There are a lot of emerging technologies coming out, AI application, deep learning based. Basically, research lab, they generate this clinical data, imaging data, it's all being generated, and then omics data. So they all collect all this information. AI does not come as such. We discussed with somebody during tea break, what is AI? Oh, there is one lady. She's here. Oh, yeah, she's here maybe. Yeah. So she's saying, oh, shall I do the AI course or something? But AI all is based on those things, right? It's all came from clinical data. AI never came itself. AI has nothing without those. So it depends on all this omics data, all this clinical data, and all those whatever it generated from that specific disease. Like for COVID, I have a lot of omics data, I have a lot of imaging data, I studied a lot of clinical. Then you make a model. So in data collections, data processing, then you make a model and then train the model. Like, okay, I made the model now. Is that able to predict what is happening? Then you just test it different way. You can stimulation models and say, hey, exactly 90% it's matching, but 10% is missing. Okay, then change the model. You have to make a better model. Maybe your markers are not, so maybe you just take if you took some of the genes and made the model, maybe you take all the genes and then make a nice model again. So that way you have to do permutation, combination, and make a nice model. Then you see the predictions. Okay, it looks like 99% I can see that model is predicting if I test it, it's going to the COVID diagnosis. Then okay. Then AI, then you can say, okay, now I have a nice model, I can use that as AI and put it there. And then anything you got the data from XYZ, any person, and put it there and then ask the model, okay, how this predicting? Is it COVID positive or negative? And then this one says, oh, based on the data you said, it does not show these symptoms, but it has this similarity, but 50% chance it may have COVID, but not. That kind of thing. It will not say, okay, it's if it is 100% matching, then it say 100%. But if that kind of data is missing, then it will give like predictions. So it's a predictive model. Remember, it's a predictive model based on the data generated from the clinical samples. See, this is another machine learning we just published. So it's a high dimensional analysis. So here also you can detect, we use for Nipah virus. We used like 29 colors, and you can do machine, meta clusters. We can talk about later. It's a little different. We have a software machine learning techniques. We stain those cells, and then do a lot of meta clustering. So, machine, the software make a lot of clusters. You can do that 1 to 26 meta cluster, and they are three-dimensional. It produces this way. And now we are comparing. So these are the colors we used. Some of them I like to say CD4, and we should have a CD8. Yeah, some of them, I don't know, 56. I don't know. We have not put 50. Yeah, 45 is the leukocyte marker, pan-leukocyte. So anyway, and then we compare. Okay, what is the difference between all metacluster and what are mostly positive, what are negative, less negative. So it gives all mathematical data. You take all the mathematical data and put it that way, all the clusters, and how those clusters related. You can do also the phylogenetic. They are more related, but they are distinctly related, all those. And then you compare. So then you can put this, and you are testing whether the Nipah virus or not Nipah virus infected monkeys, what is the difference of those clusters has a difference. In Nipah virus, this metacluster 14, the percentage of the cells go down. And then 7, Nipah virus that goes up. So that way we say, okay, so this we got a nice signature. And then you can go back, okay, metacluster 17 and 7 and 14, you saw they're different, okay? So what are the changes there? Then you can say, okay, this is very negative, this is maybe positive more. So you can get all this information. What are the different combinations? Because, okay, you say the combination permutation, in the math you have done, because it is a 29 color, and see how many combinations, several millions combination. 2 times 10 to the power 9, 29. It's a million combinations. And then so that's the machine learning does. It's impossible to do manually those different combinations. So that's why you have to do the softwares and do that machine learning and then find out, okay, this metacluster is important. And then so then we have to do that, those 2 metacluster, whether it is different from the other species or not, other infections or not, and then say, okay, this is the actual biomarker, and then you can say, okay, this is the more diagnostics. So just an example how the machine learning works. So you can also do 3D bioprinting. I think IISc have those capacity. You can make those. Use the multiple approach. You do the imaging of the tissue in your body from the human, like people do PET scan, all those. You get the whole picture of lung, whatever you want, and also you understand what material they have, kind of. You do the cell isolation. So basically, you understand the whole tissue about it, its morphology, its orientations, which layer comes first, what, and then you can use those 3D printing and make a tissue scaffold. Michigan Tech is doing that. I think IISc also has that. I think I'm talking that person. They have like 2 or 3 3D printers. They are doing all the work, and mostly it's been used for cancer therapy, wound healing, tissue engineering, bone re-engineering. So, if an army lost some part of their hand or some tissues, you can re-engineer by this printing and put it back in the past. That kind of approach is there. It's still in its infancy, but I think a lot of people are working on it to make it fine-tuned. I know the Michigan Tech has a same thing, like not the bioprinting, but they developed a kind of hemorrhagic, like if you have a hemorrhage, you're bleeding. Sometimes people die due to bleeding. You cannot stop the bleeding. So they discovered a nanoparticle, which can, if you put on this powder, just spray it on it, immediately clot, and it will stop the hemorrhages. So that kind of thing. So then again, can you introduce more cells in this, immune cells, which you lost, and that will compensate your loss of the side. So it's kind of a very model at present, and people are still doing scaffold, how to introduce different cells. The tissue damage is not only the skin, right? It has other tissues. It's the muscle cells. It's blood vessels. It has other neutrophils, lymphoid cells, non-lymphoid cells. So you have to make some kind of this thing which includes everything. So that's that. Okay, can you play the video? So this is the paper we published in 21, just to show. We can do organoids. So that's another model.
Scientists have traditionally used cancer cell lines to study animal and human biology in the lab. This has been the mainstay of experimental research for decades. However, these cell lines have limitations. They don't mimic the full complexity of an organ or tissue and a three-dimensional structure. Organoid technology has changed all that.
So it's a mini tissue.
Organoids are essentially mini tissues in a petri dish. they are much smaller than we think of an organ to typically be. In fact, intestinal organoids are about 1 to 2 millimetres across. The whole field took off when researchers in Hans Klever's lab at the Hubrecht Institute in the Netherlands identified where the stem cells were in the gut. And by taking those isolated stem cells, or small pieces of intestinal tissue, and the right growth factors, they could grow mini intestines in the lab. Every organ has its own stem cell, and by growing these with the right factors, you can make tissues from every organ in the body. To grow intestinal organoids, you need to mix the small pieces of tissue or the isolated stem cells in a matrix called matrigel, to which the cells adhere and use specific growth factors to enable them to grow into mini intestines. Here is an image of an organoid from our lab. It's about 1 millimetre across. Some organoids, however, can grow to be larger. Stem cells from one type of organ can only be grown into that organ. But if other types of stem cells, such as embryonic cells or induced pluripotent stem cells are used, you can in principle generate all the tissues in the body. We at Wageningen University are doing this for animals. In Animal Science's Complex Cell Systems program, which is part of the Next Level Animal Science theme, we are generating different organs from animals to study their biology. We are using organoids to study infectious diseases of animals and test new therapies. This will help reduce the amount of antibiotics and also the spread of antibiotic resistance. Intestinal organoids are being used to study nutrient transport, also microbe and dietary factors that support a healthy gut. This will increase the resilience of animals to stress and infection and support animal welfare. One of the other important benefits of organoid research is that they provide alternatives for the use of animals in research. And in the future, I think this will have a big impact on animal health.
Okay, so get an idea. Not this one. So organoid is an important model. So we grow 2021 also the first time rhesus macaque gut, we also call mini gut. So we took the this is the Pierce patches. From the Pierce patches, we isolate the stem cells, and you can see as the days progress, they becomes like a mini gut. So everything they have is budging, and so you eat a lot of different growth factors and all those, you add it in it. And then we characterized also like where the gut has what I grow, it's actually really gut or not. So we put different staining here. Some of them like cytokeratin is the marker of epithelial cells, which gut has also. Ki67 is the marker of proliferating cells. Those cells are proliferating. LGR5 is the stem cells. We have the LGR5 cells. HD5 is the Paneth cells. We have this. Mucin, mucus producing. Gut has always mucus producing cells. We have it. So yeah, DAPI is mostly used for the nucleus or not. So this is the, and also improved by the PCR. So that's the idea. So this is another thing to say the organoid models is also another important to diagnosis. If you have a virus, put it there rather than putting in the big animals, which may be expensive. But this nowadays, it's this is the best model. FDA has last year has said that animal model they are stopping, especially FDA. They said all the testing should be done first on the organoid model to verify. And organoid model is very important for all pharmacological industries to check the drug toxicity, and this is very good. So I'm not saying microphysiological. I saw only this slide, but I saw yesterday the Nipah virus. So this is another another model. You know, you saw that. Okay, I'll just go quickly. Okay. So basically, artificial intelligence and machine learning has an advantage, but you know, as I said, the caveat it totally depend on the clinical data. Here's some very stupid example here, whether yeah, stupid in the sense you have to. It's a simple picture, but you have a lot of information needed to make it sure. I mean, people say AI is booming, but I don't see it has a, you know, I should not get recorded here, but AI has a challenge. You know, people are talking, oh, I'll use AI, but people don't understand. It's all depend on the data. How good data you have, how valuable this data is. When you collected the data, is that data I can rely on? So those questions depends, and then you can say, especially I'm talking about the health science. Okay, I'm not talking about you know, how to go one place to another by Google Map or all those things. They can maybe very practical because the satellite is giving all the information. So this may be more, you know, good. Or if you are searching one term, you know, Wikipedia is also there. You know, the dictionary is there. So I'm not asking what is the meaning of some word, but it is the health-related issues. Like when you're talking, okay, I have the cancer or I have a COVID. Is that AI can tell me? So then the question how valid that data is, how you make that model, and the model has been validated because it's a life and death thing, right? No, when I say, tell me, you know, some doctor says you have only 2 days to live, but AI is saying, no, I have 5 more years, then who is correct, right? And so those things kind of. But again, those things are important for all the cases. Like in a, you know, here AI is including like skilled and it's replacing actually those things, but or depends on the data and how we are interpreting. Same thing like, you know, AI process of drug discovery. You know, this is going on for long. You know, we are talking to Susant also, like how they no, it's truly we're talking to Susant. It's not an example. So how they discover the drugs. You know, they, you know, all big pharmas, they do all computational now. Now, if they know the structures, like, you know, spike proteins especially, you know, you can see the tons of papers came out like 2020, 2021. Everybody looking, you know, spike proteins and doing the more conformational changes, what did happen. And people published, oh, this may be a drug of candidate. This may be the site where you can target. So that kind of things. So you can use those artificial intelligence because it is easy. If you know the structure, you can do the computer modeling and find out so many epitopes. And you can publish it also. Say, hey, you know, these are the targets. Definitely with a chemist, with a protein biologist and all those to make sure those structures are. So that's, you need the machine learning because you are using the machine learning. And then you can recommend these are the drug candidates. But traditionally, it never happened like that, you know. You know, chemical library give some targets, and then you want to see whether drug is doing something, response. Then it goes to the animal model, and then go to clinical trial, and then finally say, okay, this drug is actually working. And then it has been FDA approved or so. So yeah, that's I said, the challenges are conditions. So challenges is the data availability, AI, because it's based on how much data and the data standardization, the data quality, which is very important. It should be here actually first. So the data quality. Yeah, if the quality is wrong, the model is totally crap. Yeah, data integration and data validation. I think these two is more important. Data quality and data validations are the major key for making it AI-based model. So this is again, you know, same thing, but you know, here you can see how long it takes normally. When a drug discovery happens, at least drug you got in preclinical research, it takes 4 to 7 years. And to do the clinical studies, it takes at least 4 years, and then it is marketed. But AI can help some way because you are using a lot of modeling. So this may smooth. This is rather than spend 4 to 7 years, you may be 1 year or race. So that's that thinking in that direction, like, you know, by AI. But it also data quality, data, you know, integrity, those things is important to come to that stage. Okay, genomic analysis and precision medicine. So it is, you know, it's more important, like, you know, if what they are doing, like, you know, if you are prone or I am prone to any cancer or not. So what people are doing nowadays, it's very common in the United States also, especially people have money. They are doing their whole genome sequence and finding out how much, also you're using AI to see how much likelihood that I'll get that kind of cancer. And then that's most precision medicine. They say, okay, especially for women, like breast cancer risk, or for male prostate cancer risk, how probable it is. So people are also can do the precision medicines and find it out from the gene sequence, and it use the same AI model, put in the data whatever so have, like whatever genes people know so far causing breast cancer, whatever genes people know causes prostate cancers. And then they predict, okay, I don't find any genes, so you're less likely have a prostate cancers for the next 30 years or so, that kind of predictions. Yeah, but there are other parameters like, epidemiological, social, biological, and so these all play a role. Like, weather, climate data, you see the coverage and accessibility, it is always high because those things are readily available. So if you want to make AI model, like a weather, right? Tomorrow, how the weather will be, it will rain or not. AI can forecast much better. But when it is primary care data, health related or antibody, accessibility is low, coverage is low because there is not enough data there. So that then AI cannot do much. So this is kind of showing this. Okay. It's the same repetition, but what they do, like, this is the model they are doing. You can read that it came in Nature this year, showing that how people predict that, okay, next year there's some outbreak may come, right? Whether COVID-27 is coming or 26 is coming, something like that. So they use that same AI model. So based on what was the outbreak, okay, what was the outbreak and where's the mobility, so they can real outbreak they have, and then they do all those things. When first came, when it went, so they can make a estimations, and then they forecast. Okay, this is the now, it was like this. So forecast maybe a little go up or maybe go down or in between. So it's still they don't know, but that is the gray area. They think they may happen. So then they say, okay, the possibility of another outbreak of COVID-26 or 27 is in that range, because also include like, okay, a lot of people are already vaccinated, so maybe some way the cross protection may happen. So another COVID outbreak less likely, because everybody in this audience have a vaccine. So if I get, maybe I'll give some protection because I have some spike antibody or T cell response there. So that way they predict. Okay, basically, and then they have a model, and then they stimulate. Okay, the same thing like in United States also, like they predict like when a hurricane comes, where it is going. Is it going towards Texas or is it going to New York or where? So they use the same AI model. Okay, based on the speed it is moving, and if it, there's the water temperature is very hot, maybe it will speak up and landed as a category 5. Or if the weather, the temperature is very cold, maybe the strength will reduce and may end up with category 2. So again, they use the same AI model, but it is very easy because there are a lot of data already there, and accessibility, the satellite is there. So, data integrity, data quality is good. Okay, next, 1 more hour. We talk about vaccines. So any question about the diagnostics?
Do not walk.
Nobody? Okay, next topic is vaccine and therapeutics. So, which is important? Okay, what is the purpose of vaccine? It's written. But why? The question is why you take a vaccine? Tell me your thoughts. No, not get killed with the virus. Tell me. To COVID-19. Huh? Right, right. So you don't want to get sick so that you are protected. If there is any outbreak happens or there's somebody, somebody have a COVID, you should not, if you get it also, you can protect your body. So again, the importance is you should be healthy. So that's the purpose of taking vaccine, that you should not get infected, you'll be healthy. But how the vaccine works? Because it's an immunogen. We are putting in our bodies some antigens that induces humoral mediated antibody as well as cell mediated. But the humoral is high rated because so far the vaccines we have used in our body, we have whatever we have taken from childhood or so, it mostly induces B cell response. That's why it's a humoral, very important. T cell also does, done by cell mediated, which is, we call it like cell mediated, means T cell response. They also do that. And the delivery is done by antigen presenting cells. Okay. So is most effective way to initiate, So what are the antigen presenting cells? So the cell that display antigen with MHC complex on their surface. Probably Dr. Ashok told something yesterday, MHC, right? MHC1, MHC class 1, class 2. Huh? 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 Yeah, Redis, ready to talk about the APCs, right? Play the video. And so it will give you some idea also, like, how the antigen is presented, like if you got the vaccine or even viruses, what happens. Suppose you got a vaccine.
A key feature of the immune system is the ability to distinguish self from non-self or foreign. This remarkable ability is necessary because our bodies are constantly exposed to pathogens, for example, viruses. The basis of its ability to distinguish between both is the presentation of antigens to immune cells. Once an immune cell recognizes its specific antigen, the body launches an immune response to protect itself. Antigens are presented on the cell surface by two protein classes, MHC class 1 and MHC class 2. Let's get started with MHC class 1 molecules. Cytosolic proteins are transported to the proteasome. A proteasome is a complex that degrades proteins into small peptides of approximately 15 amino acids in length. These peptides are delivered into the endoplasmic reticulum by a Tap transporter. In the endoplasmic reticulum, Tap interacts with the MHC class 1 molecule, which is linked to several helper proteins. The term MHC stands for Major Histocompatibility Complex and refers to the molecule's importance in determining compatibility in organ transplantation. When the MHC class 1 molecule permanently binds to one of the peptide fragments, the Tap transporter and helper proteins dissociate. The antigen-loaded MHC class 1 molecule is pinched off into a vesicle and migrates through the Golgi apparatus to the cell surface. Fusion of the vesicle membrane with the plasma membrane results in the release of the antigen MHC class 1 complex. The complex can then be recognized by effector CD8 T cells. Now let's take a detailed look at the MHC class 2 antigen presentation pathway. Exogenous antigens are taken up by cells with endocytic activity. In the endosome, antigens interact with MHC class 2 molecules. The binding site of these MHC class 2 molecules is initially blocked by a small molecule. However, another helper protein present in the endosome is capable of facilitating the exchange of this small molecule with any available antigen. The vesicle then travels to the cell surface, fuses with the plasma membrane, and presents the antigen MHC class II complex on the cell surface. The antigen can now be recognized by CD4 T helper cells. So, what's the main purpose of these two antigen presentation pathways? Antigens presented by MHC class I are usually derived from inside the cell and are often referred to as endogenous antigens. These antigens can be derived from the cell, representing a normal self-antigen or neoantigens, which are derived from cancer cells. Other endogenous antigens include viral components from virus-infected cells. In contrast, MHC class II antigens are acquired from outside the cell and are therefore referred to as exogenous antigens, for example, extracellular bacteria.
So the video says only virus particle can do MHC class 1, right? That's not exactly true. Viruses also can do to some extent in class 2. There are a lot of studies has been done. There are also responsible MHC class 2 also responsible some way. It's not like clear cut, but one thing is clear cut, like yeah, it should be endosome, the virus, the agent should go inside and another is coming from the outside. So virus mostly penetrate the cells, and so that's why it's endogenous antigens, but not. But there are reports also it induces MHC class 2 also. Okay, so get some idea what is antigen presenting cells does, and those are actually primary cells are not only macrophages, because the picture shows only the macrophage, the structure like bigger cells, macrophage, but the B cells is another one, DC is another one. So they are all antigen presenting cells which presents those antigens. So how the live attenuated vaccine works, okay? Live vaccines is, we have a wild virus and you attenuate it. So attenuation can be done multiple way. You can culture it long time, and then the virus lose its pathogenicity, means it's no more aggressive, it does not do too much. Then it becomes attenuated. Attenuated means its power low down, basically. It's not virulent anymore. So weaken, mutation. So there's some mutation happens which makes the virus a little weaker. And then that weakened virus, okay, so you can see it's just a schematic pictures like, which are the virulent genes, they lost the virulent gene, that's why it's gray. So it lost the virulent gene, that means it's attenuated, okay, it's attenuated, then you can use it. And now this weak vaccine can be used for immunogen, so we can use that virus. So these are the viruses already, everybody has taken measles, mumps, rubella, varicella. These are the vaccines came from live attenuated. The one good thing is live attenuated, you don't need boostering. It's so strong, one vaccine will be enough to control it. So that's the thing, once you inject with the live attenuated virus body, what happens? It goes to the cell, it's being presented, okay? It goes to the presented to the antigen presenting cells, and the antigen cells then stimulate B cell response and T cell response. So because it is presented by antigen presenting cells, and then those antigens stimulate the B cells so that they can generate more antibodies, T cells, they can, the infected cells, they can destroy. So this is a protections, right? Already you have a good T cell response and B cells response, you can kill those. Inactivated vaccines. What are the inactivated vaccines? We have used a polio vaccine and influenza, flu vaccines. Whatever flu vaccine we have taken, all are inactivated vaccines. How they do inactivations? They kill either by inactivations or chemicals. So you have a virus which is very harmful, you killed it, and it becomes like this because it lost its pathogenicity. Um, Yesterday for KFD, Dr. Ashok mentioned like there's like formalin inactivated vaccine they have used and then they stop it. That is same like inactivated vaccines. They used formalin, a chemical, to inactivate the virus, and they are trying to use. But that side is formalin is itself a carcinogen. So there may be some trace amount of formalin left which are injecting. That's why they stopped it, because nowadays you cannot use formalin inactivated vaccine. You can use the heat killed, which is more safer, but safety is not good. So the mechanism is same thing. That virus target the immune system and antibody generates. So they mostly, it induces more B cell responses. Okay. Again, when it's common antigens, so its principle same. As you have seen the video, when they saw the antigen taken by the APCs, antigen presenting cells, presented by the MHC class 1 or class 2, and then when it is presented like this, class 1 and class 2, it binds to the CD4 or CD8 cells, right? Then everything happens. Because CD4, if it binds to the CD4, what happens CD4? That video does not say that. CD4, after binding the CD4 proliferation happens. CD4 try to produce more things, more cytokine and others. Then that CD4 also goes to the B cells and activate the B cells, so that B cells can generate that same antigens. Okay? So that antigen presentation happens, and then body create that so much B cells, okay? Presented by the cells, B cells produce so much antibody, and that antibody actually helps us to protect from the infection. Only caveat is the inactivated vaccines, because you kill most of the genes are very gone, right? Gone in the sense they're mild, the wildness is gone. That's why you may need some boosting. You have to give some boostings to make that antibody. Sometimes depends on viral vaccines, not everyone. So you do that. Subunit vaccines. Subunit, what are the vaccine? It's a recombinant polysaccharide or conjugate vaccine. So what they do, it's a viral protein. When it's called subunit vaccines, it's a viral protein. They isolated. You can do the recombinant technology nowadays. You have the protein, then you cleave those proteins, purify it by chromatography, and you have ready that antigens. And you can tag those antigen with adjuvant. What is the adjuvant is? Adjuvant means it amplifies the immune functions, okay? Adjuvant means it accelerates the immune functions. So you have target proteins, you have adjuvants, and then again it is go to the antigen presenting cells, and CD4 or CD8 binds, and then you generate so many things. Okay, same principle. We are not showing any same thing. So polysaccharide conjugate. What happened? The polysaccharides conjugate vaccine. Here also, most of the bacteria has those, those polysaccharides. Okay, so what they do, the protein conjugate is attached to the polysaccharide injector. So they use the protein conjugates, so that, because if they have polysaccharide, they easily can escape those cells, and they are virulent. A lot of bacteria does that. So if you just coat those, then they cannot escape those, and then you can present easily with the APCs, and then same thing happens. Once the cell and the cells are presented, it generates vaccines. Okay, see the list of subunit vaccines so far. I think one of the very important hepatitis B vaccines. This is the subunit vaccines, and pertussis vaccine is another. This is a meningococcus. Meningococcus. There are 4 different subtypes are there. Those are all subunit vaccines. So same thing. You have the target virus. What you do is you make it recombinant. You change some of them, and then inject. Okay. Recombinant means, understand? You have a vector, and you put that So same principle. Every vaccine goes to the APC, but only the idea is changing. When you are putting the whole virus, it's a live attenuated. When you are putting some protein, subunit. When you have the virus, you inactivate by heat or other chemicals, inactivated vaccine. When you make it recombinant, it is subunit vaccine. Okay. Viral vector vaccines. I think you guys have taken those COVID vaccines, which is a vectored vaccine. Okay. So you have a vector, like you have a spike proteins, you put with the adenovirus vectors, and now the adenovirus vector is expressing that spike proteins. Okay, and principle taken by the APCs presented, giving B cell response, T cell response. And yeah, those are the vaccine. Ebola vaccine we talked about, that was I think MVA vector. And AstraZeneca, I think it's the adenovirus vector. Ebola MVA vector, MVA, and it is adenovirus. It's a DNA virus. COVID also, there is the adenovirus vector, adeno-associated virus. Those. They say mostly the vector vaccine are very potent because why? Because the vector is always on our body, right? It does not degrade. It's multiplying, and as it multiplying, it also generating those proteins. What happened to inactivated? No question. Why inactivated vaccine you need a boosting? Anybody? No. Inactivated vaccine does not replicate. It's just a one piece because you inactivate it. It has no multiplication capacity. So once it generates, it's gone. So you have to need a boosting. But recombinant, live attenuated, the virus is always there. It's just multiplying inside. It's not bad anymore because you kill the pathogenic part. It's more milder. Recombinant is already milder because you just inserted only the target gene, and it's only producing target. So they're multiplying continuously. So that's why COVID, if you had taken the adenovirus vector vaccines, you're good because you still have the vector inside in your body, and they're producing that spike antibody continuously.
So, sir, the virus is still there in the body, but it is not.
It's a non-pathogenic, right? only you're putting that part. You're not putting the whole sequence there. Then it is pathogenic.
Is there any possibility of it mutating and becoming harmful?
No, that's nobody has. How will mutate? What will mutate? Tell me.
No, during the replication, is there any chances of it mutating?
No, it's very rare chance. Well, one thing may happen, like we talked about, flu virus, right? Flu virus, what happened? Antigenic shift, antigenic drift. But that is only flu virus characteristics. That's why for flu, they don't make that kind of vaccine. Flu virus, what they make? Inactivated vaccine or subunit vaccine. But they will not make that kind of vaccine because there are more chance of this recombination may happen. But your mutation will not be. Suppose I put this virus, now I put another same virus vector vaccine with something. But they normally don't do because it's the adenovirus has not that phenomena. What the flu vaccine orthomyxovirus does. So their principle is different. One lives in dorm, another lives in palace, that kind of thing, right? They don't talk to each other, but their principle is different, So that kind of thing. So clear a little bit. So in that case, one dose is enough, but booster may be needed. Depends on how the person's immune system and all those things. Again, so viral vector vaccine, so same thing. Yeah, so just to showing it can produce both B cells response once it's presented as well as T cell response. What about mRNA vaccine? How mRNA vaccine works? So mRNA is tagged with nanoparticles, okay? And then we inject. So what in that? So it is so mRNA, it's easier to deliver. Why only mRNA has not been given? Because if it put it as a nanoparticle, the mRNA is very, yes, to degrade, right? Very sensitive to degrade. So if I cover it, like, I have a glass bottle. If I cover it properly, very less likely I will break the glass. It's the same thing. So it's not like a hard steel, I don't need any cover. So mRNA easily can degrade. That's why they put the nanoparticle. And then they inject. And the same thing happens. It translates because it's mRNA. It's not like protein. It's mRNA. It translates. It's translated and then presented as a protein. And then same thing happens. It activates B cells and T cells. Okay. So that's the. I have taken Pfizer vaccines and BioNTech. That's mRNA vaccines. Moderna also discovered the same vaccines, mRNA vaccines. So booster response. Why the booster response is needed? So the number of naive T cells. Naive. What does the naive means? Like I never saw Rajnikant's movie, then I am naive to Rajnikant. I don't know who is Rajnikant, right? But he is a fan of it, right? He has a memory. He knows which year Rajnikant made the movie and all those. So that's the difference between me and him. So if I am a naive T cells, I'll induce to response related to the amount of antigens, okay? There are physiological limits. So I am naive and I first time to saw that Rajnikant, then I remember, okay, I should see other movies too because he's a very good actor. And so that way, then I'm no more naive, then I'm expanding. Okay, my capacity expanding. But I have a limit of capacity. I can do 10 to 15 divisions. So naive T cells can have a 10 to 15 divisions and then they take a rest because it already expanded and it generated memory. And then again, if you re-stimulate, they again make more memory. Because if you stimulate, oh no, Bapi, have you seen Rajni's recent movie? Jailer or what, that movie? Then I should see that. There's more action packed. So then my memory again, then I got a boosting and then I'm watching again. And I make more expansion of it, right? So that's the way booster works. So the same thing like when you take a booster vaccines, the same thing happens. The first dose you take, the naive T cells expand, become memory cells, and then reside. Then if you take another, I have to do something, then I expand further. And that way you have a better antibody response and all those. So check this out. There's what the CDC recommends, all the vaccines for different age and all those. So it is very nicely listed there. But the listed is for USA, I don't know India. So please play the video. So it will give some, I told, last lecture I told you what is innate and adaptive because we talk about bats' innate functions, adaptive. So give you some idea what is the difference between innate and adaptive.
Every day, we're exposed to countless pathogens, but don't get sick every time. So how does the body recognize and fight these pathogens? Let's discuss this step by step. The first physical line of defense is provided by the skin and mucous membranes. In the airways, mucus, ciliary motion, and coughing are important defense mechanisms. In addition, bodily secretions also protect us from pathogens. For example, saliva contains lysozyme, which can destroy the cell wall of gram-positive bacteria. If we take a look at the stomach, gastric acid creates a hostile environment for most bacteria. Also, the normal flora residing on the skin and mucous membranes displace and fight pathogens. If the pathogen ends up breaching these barriers, the body depends on further active defense mechanisms of the immune system. First, the cells involved in innate immunity become active. Certain stimuli, such as tissue injury, trigger an inflammatory reaction. As a result, leukocytes, especially granulocytes and macrophages, are attracted to the site of damage. They leave the adjacent blood cells and penetrate into the damaged tissue. The body recognizes certain specific repeating patterns present on many pathogens as foreign. These include, for example, lipopolysaccharides, which are located on the outer membrane of gram-negative bacteria. These characteristics are also called pathogen-associated molecular patterns, in short, PAMPs. Because they're commonly shared between different pathogen subtypes, they're considered nonspecific. The macrophages and granulocytes attracted by the inflammation recognize the PAMPs of pathogens through pattern recognition receptors, in short, PRR. The granulocytes then secrete a toxic substance to fight against the invading bacteria. The macrophages engulf the bacteria, or in other words, phagocytose them. In addition, PAMPs activate the complement system. This system consists of many different proteins that initially circulate in the blood and tissue in an inactive state. These proteins are activated by pathogens, leading to a cascade of reactions. This leads to three effects. First, more leukocytes are attracted to the site, which stimulates the inflammation. Second, the complement components mark the pathogens, facilitating phagocytosis, which is required for some pathogens. Third, the complement cascade leads to the formation of the membrane attack complex, in short, MAC, which produces pores that especially kill gram-negative bacteria. As well as the innate immune system, the adaptive immune system is also activated. Its reactions start at a later stage, and the response is initially slower. Through its high degree of specificity, the adaptive immune system can efficiently fight against the invading pathogen, preventing it from spreading. Antigen-presenting cells, such as macrophages and dendritic cells, represent the intersection between the innate and adaptive immune systems. These cells take up microbial components, known as antigens, process them, and present them on the cell surface via MHC molecules, which stands for Major Histocompatibility Complex. As a result, the adaptive immune system is activated. How macrophages and dendritic cells present antigens can be seen in our video on antigen presentation. The decisive cells of the humoral immune system are T and B cells. They circulate in the blood, but are also present in lymphatic tissues such as lymph nodes, tonsils, and intestinal mucosa. Their immune response is triggered by antigens. T and B cells become activated upon contact with the antigen specific to their receptor. Let's first look at T cells, which comprise two main groups. T cells are divided into two subsets based on their surface proteins: CD4 positive T cells and CD8 positive T cells. CD4 positive T cells are also called T helper cells. They activate macrophages and granulocytes or help to activate B cells. CD8 positive cytotoxic T cells kill virus-infected or tumor cells. Now, let's take a closer look at CD4 positive T cells. The dendritic cell presents the uptaken antigen on its surface via MHC class II receptors to CD4 positive T cells. The corresponding CD4 positive T cell binds via the antigen-specific T cell receptor and other auxiliary proteins to the dendritic cell. The release of chemical messengers, namely cytokines, induces CD4 positive T cell proliferation. Depending on the pathogen, the CD4 positive T cell differentiates into one of several subtypes. These principally develop into different T helper cells, regulatory T cells, and over time, into memory T cells. A special group is the follicular helper T cells, in short, TFH cells, which are important for B cell activation. We'll get back to them again later. Regulatory T cells suppress an excessive immune response. Memory cells remain in the body for years and elicit an immediate adaptive immune response to subsequent exposure to the same antigen. Depending on the invading pathogen, other T helper cells activate the cells of the innate immune response through cytokines. In addition to CD4 positive T cells, CD8 positive T cells can also be activated. In contrast to CD4 positive T cells, these cytotoxic T cells are activated by MHC class I antigen presentation and not MHC class II. Cytotoxic T cells recognize virus-infected or damaged cells and induce programmed cell death, also termed apoptosis. Now, on to B cells. These cells can fight off pathogens with antibodies. B cells can recognize antigens in one of two ways. Like T cells, they can detect presented antigens. Also, they can recognize antigens directly without antigen presentation. Once a B cell finds the antigen matching its receptor, it's then presented via MHC class II on the cell surface. If this antigen is recognized by a matching, previously formed TFH cell, it then binds to the B cell with its T cell receptor and auxiliary proteins. After this double check, the B cell is activated by cytokines and starts to proliferate. As a result, memory and plasma cells are formed. Both memory B and T cells are responsible for a rapid immune response to reinfection. The plasma cell produces antibodies that match the antigen on the pathogen. Initially, IgM antibodies are formed. After interaction of the plasma cell with the T helper cell, high-affinity IgG antibodies and other antibodies that are adapted to specific needs are generated. The antibodies now bind to the invading pathogen. The final stage of the adaptive immune response comprises three parts. First, antibody binding can render the pathogen harmless. Second, this binding results in more efficient phagocytosis through opsonization. Third, the complement system is reactivated. As mentioned previously, this enables the pathogen to be directly lysed by the membrane attack complex. However, other leukocytes are also attracted, leading to opsonization of the pathogen, thereby facilitating phagocytosis. Let's summarize the most important facts about the immune response. The innate immune system provides a rough recognition and rapid first line of defense to invading pathogens. Antigen-presenting cells activate the adaptive immune system and establish a link between the innate and adaptive immune systems. Through antibodies and cytotoxic T cells, the adaptive immune system enables a highly efficient, although late response against the invading pathogen. Because of its immune memory, it elicits a highly rapid response upon renewed infection. In this process, the complement system plays an important part in two respects. First, it's involved in initiating the innate immune response. Second, it's part of the common final pathway of the innate and adaptive immune responses. So, innate and adaptive immunity are interlinked in many ways. Whether it's fighting the coronavirus or the common cold, the body's immune system has several mechanisms to combat viral infection.
Okay, so adjuvants we talked briefly, like well, this is the additive to vaccine that enhance immune induction. So, we talked about subunit vaccines, like you have the antigens and tagged to the adjuvants. Basically, because that's small, tiny, so sometimes it's better to present with the antigen, or sometimes they put to the carrier proteins so that it better presents and induces more immune functions. So that's why the adjuvants also can bring professional APC contact with antigens through pro-inflammatory reactions also, and can drive stronger cell-mediated antibody. So basically, adjuvants means it accelerates the antigen presentations, better B cell response, T cell response, those things, the function of adjuvants. And also, that's more important, can stabilize antigen and extend presentation of it in the blood. So it does not allow to degrade easily, so it stabilizes the antigen so that it presented better. So there are a lot of adjuvants has been used so far. Those red colors means those are approved for humans. It has been licensed, but the others has been tested. Yeah, so nothing to talk much about it, because this is only you can remember, like for mRNA we use the nanoparticles. and the lipid nanoparticle is one of the adjuvant. That's why the mRNA is one thing is mRNA is highly degradable, but the nanoparticle actually it stabilizes. And the function is adjuvant's function is stabilize the antigen, so it protects it, and it's also adjuvants and used for mRNA vaccines. So this is the mission of 2030. So for example, surface immunoglobulin, surface soluble hepatitis B antigens, okay? It's a hepatitis B antigens when normally we used with alum adjuvants, it's a adjuvant, we talked the previous slide. And with the antigens, we put as a vaccine. But what happened, normally 30% are unresponsive, okay? Though we think that vaccine will induce immunity for everybody, it does not. So what's the cause? Maybe we can think about, we are not giving enough antigen maybe, or the antigen presentation not happening, right? That's the major reason. Somehow antigen presentation not happening. So recently, that is the alternative, and it has been applying for, I think, couple years now. So what they did, they increased the dosage. Whatever they did, they had 3 to 4 dosage. Now they have increased the dosage, and also they have changed the adjuvants. Okay, and so also thinking that they gave additional antigens. So basically, that antigen is not covering enough. So you give more antigens, better adjuvants, so that you have more. So at least they found still some part is unresponsive. It's covering maybe 75% or maybe 80%, but some part is still unresponsive. So this is the future of what they're thinking. So hepatitis B has been discovered long ago. Vaccine has been discovered long ago, but still, and I'll come the next slide, how much prevalence of the hepatitis B, but still it is not, the vaccine is not protecting everybody. So that's, people are now thinking, what's the reason? So that is the potential future strategy. This has been applied right now. More virus, more antigens, different adjuvants, it has been applied right now. This is the potential future. So what they are thinking, been given by intramuscular, so it is given to the dermis, okay? So it is now they are thinking to inject on intradermal, where there are more Langerhans cells, the disease are present, intradermal regions. And also they are thinking to give you a microneedle, so it should be like some kind of come this, and it is a microneedle, and what they will do, they patch on you and the intradermal regions, and all these needles has containing the vaccine, and so it will be like this, and you get multiple exposures in a place, and also going intradermal, and it also takes less vaccine than what normally was given before. So you're not giving too much. It says 1/5 of it you give, and that way they have found out some preliminary studies. It is giving much higher immune functions, immune roles, and kind of 99% people has, but it's trial. It has not been, finalized yet. It's still under experimental conditions. So, but they found more persons which you follow that kind of procedures, they generate those responses early. So prevalent case of chronic hepatitis B by WHO 2022, you can see here. This is the Asia, this is South Asia. So this is the range, how many people are infected, okay? And so you can see overall, everywhere the hepatitis B is there, right? Though the vaccine came long ago, still we have infections. This is the chronic hepatitis B. That means there is cirrhosis. So the chronic hepatitis causes cirrhosis. Your liver is harder. Liver is one of the organs which can regenerate easily. If you cut a liver, little piece, it come back, because liver, only the organ liver has that functions. But when it becomes chronic, it becomes solid. So that the liver can be done until you do the liver transplantations or you are dead, because the liver is the major organ which filters a lot of impurities from the blood. So basically, you're showing there a lot of prevalence there for the hepatitis B. Same like the hepatitis C. You can see everywhere, and it's a huge number, some millions. All those in millions, there are hepatitis C's there. There are hepatitis B, C, where I think drugs came out something like 2014 or so. It's a Gilead, I think, made them. It's a very good drug. It's a combination of drug. It can cure, but that is for if somebody exposed to hepatitis C, you just give the drug that. But here, already have chronic stage, that drug may not work because you're already late stage. But you can see how much hepatitis C prevalence is present throughout the world. And he said the progress toward global viral hepatitis target. You can see these are the number of hepatitis B per year, targets, progress, number of hepatitis C infection per year, percentage of people who inject drugs, number of people dying from the hepatitis B, so these are the very important, see these figures. Though the vaccine, vaccines, it's 0.82, now 1.1, but still, the 530,000 people are dying. I don't know, maybe due to the not vaccine coverage, or the vaccine is not efficient enough to, we saw the couple slides before, like the way vaccine is, and it's not giving protection. So one of the key reasons, definitely the way it's administered is there, and maybe another is like not everybody takes hepatitis B, and that's why I take the hepatitis B, because it's a highly prevalent vaccine, disease, and vaccine is very effective on this. So nanovaccine, just giving a glimpse of how the nanovaccine, seen already from the video how adaptive and everything is the same principle like for mRNA vaccine. mRNA is a nanovaccine, like mRNA nanoparticles and nanovaccines. So this is the techniques people are now using the nanovaccine administration. How you can do this is that goes to the injection. You give the injection, the same thing. It's prevented by the disease. You saw the video, PAMP and DAMP. So that is the pathogen associated microparticles, and that has been bind to the PAR and PAMPs. They bind together, and then it's internalized by endosomes, and then it's been presented APCs, and then it's, you saw the video, bind to the CD4, CD8, then the MHC class triggered with the CD4 goes to the B cells, and B cell also gets self check by both way with the soluble antigens as well as the CD4 binded antigens, and then produce an antibody. Okay, so there are multiple kind of low affinity antibody, high antibody, those are theirs, and also the CD8 activated T cells are there. They are also have the killing functions by killing those cytokines. So that is the principle of nano. It's the same, like it's the same. It's not like, don't think about it's different than the live attenuated vaccine. It's same, but nano vaccine is the new principle, new technology. It's very popular now after the mRNA vaccines, and while different are now under investigations. People are doing like polymer, lipid-based, cell-assembled, biomimetic, and inorganic. So people are trying different things. For example, here, there is a polymeric adjuvants they used as like, for example, hemagglutinin proteins. You can take maybe it's from flu virus, right? Hemagglutinin, they have hemagglutinin neuraminidase. So you just make a polymer, but it's still in different investigational stage. All those are concepts. So they are trying to make those polymeric nanoparticle vaccines in different way. And those are those. So far, US FDA has approved those vaccines, and I mentioned like Moderna, Pfizer's, those are mRNA vaccines, the nanoparticle vaccines. But there is also some vaccines for hepatitis B, Hebrio and Engerix, those are hepatitis B vaccines are there. The first one is, that was the recombinant protein vaccines. I think A lot of people takes those recombinant vax, which is also nanoparticle vaccines earlier. But these are already approved vaccines, but there are so many other nanoparticle vaccines are under clinical trials. But there are challenges. So challenges in the sense, I still think that the mRNA vaccine for Pfizer is still under investigation, how the antigen presentation happens. All not exactly clear how the innate function comes fast, those things. So nanoparticle receptor interaction with immune cells, it's not clearly known. It still need to be studies more, and how the uptake by APCs and all those things. Those are still hypothetical we put together. I think there are more to be side. Translatability of those therapeutics for small-scale synthesis to scale up production for commercial, because still those particles still in a very scale stage, to find out whether they are not itself becomes a antigen and do something wrong. So those are another thing. Nanoparticle, make sure those are not like people say microplastics, right? You're giving a lot of nanoparticles, it should not create some other safety issues and all those. That's another thing. So that's why there is a lot of we have to do long-term research and see how it has any long-term effect or not. So drug development strategies, there's, we talked about drug repurposing, those things, but these are common drugs, and it has been approved for different viruses like influenza. Those are the drugs always used. Tamiflu is the oseltamivir. HIV, those are the drugs always used. Hepatitis B, also these. So these are already marketed. Acyclovir for herpes simplex. But this is very limited. Only for certain viruses, these drugs works very good. And these are the mechanism of action where they actually affects those, which enzyme or which pathway they act on. So based on viral protein targets, it requires detailed understanding of virus. Before you do the drug discovery, you should know the replications and what you can target in, like KFD, like carcinoid disease. He presented, and I also shown like these are the pathway. Even the pathway is not clearly known because we don't have laboratory either. So those things need to be studied further. And then you can target some drugs, maybe doing the repurposing of those which are already known, because that is also RNA virus, this is RNA. So can I try those, and can I target those? Yeah, that's the kind of thing here. So for HIV, the replication is well known. So you can target those. Okay, can I target the wiring? Can you stop the protease when it releases? So protease inhibitor you can do. When it is doing maturation, can you stop here? Basically prevent tree, those things. So this is a good example because HIV replication has been studied so much. It's more clear where the proteins are producing, what step they can work on. So that's why HIV is very good too. There are a lot of drugs, you saw that chart, right? And the more important, so always they use the integrase, reverse transcriptase. These two should be there because they are the major pathway, reverse transcriptase and integrase. And the other, sometimes they use the entry inhibitor. Entry inhibitor not that much, but the protease inhibitor is very important. So.
Which target is more effective here?
So for HIV, they use the cocktail, right? Yeah. So not a single target. So they use this, and mostly this. Yeah. But sometimes the new drugs, which takes, I think, once in 6 months, everything. Yeah, they don't want to take any risks. So also you can target the host cell where it is replicating rather than going to, so indirect approach. Rather than going in the virus, you can target the host cells. But only the point is like, non-specifically you may kill also good cells too, right? So adverse effects on normal cell function may be problematic, particularly for chronic viral diseases. So it has both pros and cons there, to target the host cells that way. So these are the drugs used for the, HIV, like Miravirk, block CCR5 receptors. CCR5 and CXCR4, those are the co-receptors used for HIV for virus entry. Okay, so Maravirok, so that virus does not get in. But the another caveat is that you're blocking CCR5 also normal cells too, which is not infected by HIV or going to infect it. So there is maybe some side effect. Always block some receptors unspecifically or non-specifically, may have sequence adverse reactions. So that's why there are more chances. So you can see some has been approved, but a lot of things are under research. Same those drugs for Ebola also. It not used generally those drugs because monoclonal antibody, they have vaccines. Monoclonal antibody is given, which is more safer. But those, they can block some of the tyrosine kinase pathway, which is always present in normal cells too. So we don't know what is the impact will be if you block those pathways, and so it has limitations, those things. Okay, so viral pathogens and antiviral strategy. Acute disease is difficult and arguably not effective at controlled disease, because this is due to the prophylaxis. Persistent viral infection are attractive targets, accurate diagnosis of infections, time to evaluate prior to initiation therapy. So you can do two different strategies. One can reduce mortality and transmission even if it not cure, or facilitate host immune response to clear virus infections. Understand? It's very simple. So acute disease is very difficult to control, but when it's and all those, then you can plan accordingly. So drug repurposing which is already been established. You have seen that later virus, those drugs, hepatitis B, hepatitis C, the drugs are already marketed. So now disease like this is RNA virus. This is DNA virus. Can I use those against drugs? So that's the idea, to explore that drugs for other disease rather than spending money for discovering new drugs for treating those diseases. So these are the drug repurposing in use. So many drugs, there is not too much drugs, but other drugs being used to treat it. Chikungunya, so it's kind of example like dengue virus. So many drugs has been used either for cancer therapy, bacterial infections, viral infections. They focus on polymerization, capsid binding protein, or those, they target those proteins. So if they are known already, those drugs has been already shown, though, okay, they just work on some way RNA dependent RNA polymers or DNA dependent DNA polymers. So then let's try it and give a shot and see if it works. Sometimes it works. Some COVID, ribavirin, and other drugs, I forgot the name. So yeah, people have tried those. So play the video. I think that's most, there is nothing to talk more on repurposing. It's kind of trial and error. They have not done too much on those.
How do you recognize a visionary decision? difficult because it was relatively densely populated, and it also had the worst.
When you think about virus and diseases and you think about treatment, it really is a war. It's a battle for survival. It's a game of move and counter-move.
And that's why smallpox is such an important virus, because it did show people that you could do something about these things. These weren't visited upon us from an evil god. They were viruses that you could defeat with science.
The whole story of the World Health Organization's smallpox eradication campaign, I think, is one of the most amazing and moving stories in 20th century medicine.
This is one of those great endeavours where human intelligence and goodwill across the world came together.
When the eradication program began, we were losing 2 million people a year to smallpox. And remember, only a third of the people died. A lot of other people horribly scarred, and then many of them were left blind.
At the time the smallpox eradication program started, there were 5 regions of the world that had smallpox. South America, mainly Brazil, West and Central Africa, East and Southern Africa, the Indian subcontinent, and Indonesia.
In 1958, at the World Health Assembly, it was proposed by the Russians to eradicate smallpox. This is the first time the Russians had been in the World Health Assembly for years. So all the countries said, okay, this is what the Russians want, let's keep the Russians happy, and so we'll approve it.
Nothing like the eradication had ever been attempted before.
The rumor is that then Director General thought for sure this would fail, and so he decided, let's make the head of an American. Then when it fails, we can pin it on this guy Henderson.
In the early 1960s, I was in the Centers for Disease Control, and my name came up to go. I didn't want to go.
D.A. Henderson got dragooned into going to Geneva and running the smallpox eradication. And he scratches his head and tries to figure out, okay, you know, how are we going to do the equivalent of a moon landing with this virus?
It was plunging into deep waters that take this on.
They started with 23 countries in Africa that were known to have smallpox.
Our strategy was to vaccinate the entire population. You're talking about hundreds of millions of people. At one point, we had 6,800 outbreaks, and each outbreak could be hundreds of cases. Even in retrospect, it seems impossible to deal with 6,800 outbreaks of smallpox at one time. But we believed it could be done. This was a disease with a very good vaccine, and you can't hide it. If a person gets smallpox, you're going to be able to find them.
West Africa was deemed to be the most difficult because it was relatively densely populated, and it also had the worst infrastructure.
In December of 1966, we got notice of a smallpox outbreak in eastern Nigeria, but we didn't have a vaccine. We had a little bit, not enough. So we couldn't do mass vaccination. So what we did was we used a network of missionaries who would get on the radio every night and report new outbreaks. And with a map in front of me, I divided up the area to ask each missionary to send runners to the villages in their area the next day. 24 hours later, I knew exactly where the smallpox cases were. And so we were able to take our small amount of vaccine and go into those villages. We vaccinated them, and the outbreak stopped so fast that it took our breath away.
So Bill Fahey stumbled upon the notion of what is called surveillance and containment, or ring vaccination. And it happened because they simply ran out of enough vaccine.
They just vaccinated around the cases, and lo and behold, bingo, it disappeared because the virus had nowhere to go.
In one sense, we outthought the smallpox virus, which shouldn't be hard to do when you realize they don't have a brain. But that's what we were trying to do. One by one, other countries were able to accomplish this. And by 1973, India was the biggest problem left.
In 1974, India had about 500 million people. They lived in about 120 million houses in about 500,000 villages in 21 states. And to eradicate smallpox, it meant that we would have to visit every single house in India. And ultimately we did. We visited 120 million houses in India every month for about 20 months. We did more than 1 billion house calls.
It was a crusade. They were inspired, not only by one another, but by the fact that, my goodness, we can actually get rid of a disease.
We chipped away at it with our Indian team until we were able to show that we were containing more outbreaks than we were getting new ones. And that was the tipping point.
It looked like it was coming together, and it was in February of 1974 that finally decided with Bill, we can do it.
I think that last year was probably the most incredible public health story that I have seen in all of global health history.
Across the world, we had managed to go from 10 million cases and 2 million deaths a year in just 10 years down to 0. You feel pretty good about it.
Smallpox eradication was one of the greatest accomplishments of the human race.
I'm sure there are literally billions of lives that have been saved as a result.
Good news from Geneva. The World Health Organization has made it official. Smallpox throughout the world has been virtually eliminated.
After eradication was declared in 1980, there were still samples of variola virus around the world. One accident, one confirmed case is a pandemic. Smallpox was still a threat.
So this is the end of my talk. The point is this topic impossible. It has so much difficult to replication. Who has time to do that? It will take so much time. Which lab is doing those things? But there's some way to do it. I mean, that's the simple way. Smallpox, when it has been eradicated, where is the fancy lab? Where is all those special techniques and all those? I think it's a dedication and thought process, right? Okay, can you do this way? And people find out the alternatives, same like a COVID, right? And we quickly came out some vaccine and other recombinant in modern era. And also earlier, like 1975, it has been eradicated. So it is possible. Whether we challenge it with any viruses or in future, I think we should have that kind of thought process. I think that's more important, like how you challenge those. And there are a lot of protocols we see, is lab attenuated, which lab attenuated takes time. You have to adapt, and then see, and also measure, like, it's got attenuated or not. But definitely subunit, inactivated, those are easy. People that way did, like, that take the virus, maybe boiled it, heat it, and then it inactivated, people injected it. Get some protections. And another thing is the monoclonal antibody. Like, if somebody survived, collect the monoclonal, the antibody, plasma collected, use the serum to, which is called convalescent plasma, and inject that person who is infected. Some way you get protection. So that's pretty much. And any questions you have on vaccines, therapeutics? No question? Oh, yeah. So, okay. So, subunit, so recombinant. Recombinant means what? You need a vector. Okay? You need a vector and then put that antigen in that vector. That is the recombinant. Subunit can be a small peptides, okay, like spike protein. I somehow isolated that peptide and I can inject it. That's the subunit. It's a small protein. But here recombinant is you are inserting within the gene of that recombinant virus. Normally people use recombinant virus. So like for example adenovirus vector, there are pox virus vector, there are herpes virus vector. These are the very popular vectors people use. Oh, they are also used what is called rabies virus vector. We studied also the rabies virus. So those are the vector. So what they do, that vector, they take out all those bad genes, pathogenic, which causes death. They take out all those bad genes. And like I know, like for example, I'm taking the example COVID. I know the spike is very immunogenic. Immunogenic means it will produce good antibody, good T cell response. I take out those bad parts from those from rabies vector or adenovector, and then put that spike to that virus and make a construct. That's why the biotechnology, molecular biology, they do that kind of, they always do this recombination, all those things. And now this is more safe. It is not less pathogenic for rabies also, and only have spike protein. It is also less pathogenic for COVID. So it is totally less. So that can replicate inside. And when you inject it, replicate inside, it produces always that protein. It's a different, yeah.
Any other question? So then,
like I
yeah.
once like information like all to their age, they have the idea like what vaccines they have taken in their childhood. If they have that list with them, so what is missed or anything it is we need to take in care, because recently there was rabies when like dog bite generally. So we saw many people die during dose compliment.
Right.
So what suggestions can we get with respect to?
So yeah, so that in one of the slide, I think couple slides ago, I saw that CDC has recommended what is United States. I should follow that, because that is CDC guidelines. CDC, WHO, they always collaborate. They are always talking the same thing. So you should follow that link, CDC website, and it is very detailed. If he's immunocompromised, it is between very newborn, on what age, adults, everything listed, what vaccine need to be done. And if you
mandatory, I think it's made mandatory to take all these vaccines which have been listed out there.
it's listed out there, yeah. It's listed there, and you just say check with yours. And they check with your whatever you've taken, and they say, okay, and especially it's important, like, if you are traveling, right? If you're traveling outside country, the country where there may be more yellow fever or some outbreak happened. So you also have to be always little proactive to see, if I go to the Japan, because Japan recently has declared that flu outbreak. They said in the newspaper. So if you go, definitely you should take flu vaccine before you go in. So those things you also have to keep in, like United States, like when I came, so we always get an email, like especially because I'm traveling to internationally, the email come from my university as well as from, they say check the link. So there is a travel.gov, it's a website, and it always update every time. So those are traveling in this country, this vaccine is mandatory. They say it's better you take it. You take it. If you are traveling Africa, better you take this vaccine. So they always forecast. Even they say, for me, they say, don't travel Jammu Kashmir, this thing. I mean, those other aspects also, these are not safe. Yeah, but also they also talk about my health. Okay, if you're going, have you taken this vaccine? Even they told me, have you taken flu vaccine, because they're going. So those are the recommendations you have to follow too. I don't know about the Ministry of Health in Government of India, but I think they should have something like that kind of framework to inform. But if you want to do that vaccine list, I should recommend go to the CDC website. They have a very nicely detailed information there. So
make it mandatory again. These vaccines are going to be little too expensive. In case if it is freely available, then I think probably they can make it mandatory.
Well, yeah, yeah,
That's the end user.
right.
So some of them tell that viral mutations, is it a part of evolution or something?
Of course, yeah. So that yeah.
There was a lot of discussion there. It was engineered in one of the labs in China, but again, most of the evidence they indicate that it evolved naturally.
Right. I mean, I should not talk about COVID, but but other yeah, that's another controversial area. I mean, there are a lot of theories and all those things. I think we'll never able to understand what happened, right, that time. But if you are asking the evolution question, yes, especially RNA virus, those are segmented, those are negative strand, they always mutate. And especially if you have seen the history of HIV, you know, it mutates. There is no vaccine still, and I don't think there will be, because that kind of virus mutates all the time, and it's very difficult to control those. DNA virus is little more stable because it's double-stranded DNA. They don't mutate too much, but they are very less. Only one was that smallpox, you know, but they able to carry because the smallpox does not have too much variant. That's why the vaccine works very good, it's clear. But others it's challenging. But that's why you have to monitor. Like you probably saw that measles outbreak happened, right? Even Mpox, it was there before. It's not new virus. So it's re-emerging happening, right? And it's a new variant. Then okay, I'll go for it and check for it. If the vaccine works, that's good, you know, those things. Yeah.
Have a discussion there over lunch. Anybody has not given feedback form? All of you given? Feedback.
And thank you so much for your patience and interest in this, and I really enjoyed it for the last 5 years.